Hello, my name is Erin Reddy, and presenting with me today is Erin Marker. Together, we worked on this project in Dr. Raina Robeva's Honors 131 Intro to Systems Biology class. So, without further ado, let's talk about bistability of the lac operon. This is a very brief overview on the biology of the lac operon, focusing only on the main parts relevant to our project. The lac operon is responsible for metabolizing lactose and it is inducible, which means it is only activated under certain conditions, those being when lactose is present and glucose is absent. Then we have listed the 3 genes contained in the lac operon and the enzymes they produce. However, in this project, we are only concerned with the LAC-Z and LAC-Y enzymes because little is known about LAC-A and its necessity in this whole process. As you can see, we chose not to mention catabolite repression as it does not have a significant impact, and we really wanted to show that a minimal model can still very well capture bistability. Here is a diagram of when lactose is not present and the operon is off. The repressor protein is active and blocking transcription of the LAC-Z and LAC-Y genes. Now here's a visual of when lactose is present and the operon is on. The repressor is inactive, allowing for the transcription of the LAC-Z and LAC-Y genes. Also, I'd like to note that LAC-Z and LAC-Y are produced simultaneously. So later you will see in our equations that we grouped them under one variable instead of 2 because they are either produced together or not at all. Moving on to the math side of things, my personal favorite, we have here the basics of Boolean models. Boolean models are a way to mathematically explain certain biological systems and processes. There are only 2 possible values, 1 representing high concentrations or 0 representing low concentrations. There are 3 main operations in Boolean functions, and, or, and not. We provided examples to help in understanding what each of those mean. As for update functions, those along with the initial condition of the variables dictate what the values of the variables will be in the next time instance. We also wrote a general update equation. We will have an equation for each variable. So to illustrate the transitions between the states of the system, we used state space diagrams. Each state is represented by a sequence of zeros and ones to describe the state of each variable. The most important part to focus on in a state space diagram is arguably the fixed points of the system. In the example figure shown, the fixed points or steady states are 0, 0, and 1, 1, and they are signified by the revolving arrow. The fixed points are the outcomes of the model. For the lac operon, they will correspond to the on and off states. Now we have some general background on bistability. Simply put, bistability is when the same external conditions can result in different steady states. As for the lac operon, when there is a medium level of external lactose, the system can be either turned on or off. It is important to note that bistability is biologically advantageous as it allows for quick transitioning between states in response to an input, resulting in an all or nothing behavior after a certain threshold is crossed. The value of said threshold depends on the system's history. And bistability is in no way limited to the lac operon. It has been demonstrated in several other systems, such as the pathophysiology of some diseases and in cell differentiation. This figure shows the original update functions we were given for a minimal model of the lac operon. In this model, we only had update equations for 3 variables, mRNA, the enzymes, and internal lactose. I'm going to quickly translate the update functions into proper sentences to help in understanding what they really mean. So for the first update function, to have mRNA in the next time instance, we need to have no external glucose and internal lactose or external lactose. Next, to have enzymes in the next time instance, we need to have mRNA now. And finally, to have internal lactose, we need to currently have no external glucose and enzymes and external lactose, or internal lactose and no enzymes. As you can see in the figure on the bottom, we added equations for external glucose and external lactose to accurately account for all the necessary variables. We had these variables equal themselves because they update at a much slower rate than the other variables. This means the values of these variables will stay the same in the next time instance. So again, to translate them, it would simply be to have glucose in the next time instance, there needs to be glucose present now. And to have external lactose in the next time instance, we need to have external lactose now. Below the update functions is the state space diagram for the model. These update functions produced 4 possible steady states that the system could result in. Again, the steady states are signified by the revolving arrow, and this just means the system will remain in that state for all of the following time instances. I'll go into further detail of these steady states on the next slide. So here we have the 4 steady states again from the model on the previous slide. The last 2 values in each sequence correspond to the external conditions of the system. So 0, 0 means the absence of both external glucose and external lactose. 0, 1 means glucose is absent and lactose is present. 1, 0 means glucose is present and lactose is absent. And 1, 1 means both glucose and lactose are present. Of the 4 possible steady states, the model is only turned on when external glucose is absent and external lactose is present. Although this is a biologically feasible minimal model, it does not reflect bistability, as there is no consideration for intermediate levels of lactose, and there is only a single fixed point for each of the external conditions. So here's where our main question comes into play. Can we make a modification to the minimal model so that it will result in 2 fixed points, on and off, for intermediate concentrations of external lactose and thus demonstrate bistability? Now Erin is going to discuss how we did this and what we found.
Thank you. So in order for the model to be able to show bistability, the addition of another variable is required. The variable that we decided to add was a variable representing high levels of external lactose. This will capture an important aspect of the lac operon and to enable it to show bistability. So when both the variable for LE and LE high are both equal to 1, this will represent a high level of external lactose. When LE is equal to 1 and LE high is equal to 0, this will represent an intermediate level of external lactose. And when LE and LE high are both equal to 0, then this will represent a low level of external lactose. Bistability will be shown if when there's an intermediate level of external lactose, if there are 2 different outcomes that are possible. When creating a more accurate model of the lactose operon, it's important to consider the volume of external lactose present. If there's a medium amount of external lactose present, then the lac permease is required to bring the external lactose into the cell so it can be used for the production of enzymes. However, if there's a very large amount of external lactose present, it can be assumed that there are trace amounts of lac permease still in the cell to bring that lactose into the cell. There are 2 main additions to the update functions that we came up with to account for the level of external lactose in the system and to demonstrate the bistable behavior that the lac operon shows. These changes are shown in Figure 7. The first modification to the update functions is to the update function for mRNA. Now, in order for mRNA to be produced, there needs to be no external glucose present and either lactose already present in the cell or a high level of external lactose present. This demonstrates the fact that if there is a high level of external lactose, it can be assumed that there are trace amounts of lacpermise to let the external lactose into the cell. The second modification is to the third update function, the update function for lactose. In order for lactose to be present in the next time step, there needs to be no external glucose and enzymes representing lacpermise and external lactose, or high levels of external lactose, or internal lactose and no enzymes to break down that internal lactose. This shows the same idea that was presented on the last slide, that if there are trace amounts of lacpermise and a large quantity of external lactose will still make its way into the cell. With the addition of the variable to keep track of high levels of external lactose, there's a state that it's possible where external lactose is equal to 0 and high levels of external lactose is equal to 1. This does not correspond to any true state, so in order to remove this state, the update function for high levels of external lactose is modified so that high levels of external lactose will exist in the next time step if external lactose and high levels of external lactose are both present now. Figure 9 shows a state space diagram for the modified update functions. These steady states are shown by the nodes that have an arrow that feeds into themselves. Figure 10 shows a table of the steady states of the modified update functions. In the first 3 states, glucose is present, so no enzymes are produced and the lac operon is off. In the last 4 states, glucose is not present. In the 4th state down, glucose is not present, but external lactose is low, so no enzymes are produced and the lac operon is off. In the 2 rows highlighted in blue, external lactose is at a medium level. In one case, the lac operon is on and enzymes are produced. In the other case, the lac operon is off and enzymes are not produced. This demonstrates the bistability behavior of the lac operon. In the final state, glucose is not present and external lactose is at a high level. Enzymes are produced and the lac operon is on. In introducing a variable representing high levels of external lactose to the model and modifying the update functions for messenger RNA and lactose, we have shown that the model for the lactose operon demonstrates bistability.
Finally, here are our references. Thank you so much for taking the time to watch our presentation. We hope you enjoyed listening as much as we enjoyed working on this project. Thank you.